Let's consider a hypothetical gene. I will take a small region for demonstration and I will ask a few questions now. Is this gene expressed in the cells of normal skin? Is it also expressed in the skin cells of cancer patients? If yes, what is the relative amount of the RNA produced? Well, I don't know what is the situation in the tissues. The transcription might be occurring and different quantities like this could be produced. We don't know what is the situation. We would like to detect, but how? Well, we can use the complementary nature of nucleotides. We know A pairs with T, G always pairs with C. This type of complementarity causes hybridization of DNA and RNA strands. I can use that. That means I can design probes. If I identify unique regions in the mRNA or cDNA, I can note down the complementary sequence and synthesize them in large quantities and use these molecules, probe molecules, to track the sequence of our interest, the molecule of our interest. If this is done correctly, the hybridization will be very specific. We can prepare probe spots based on this principle. That is, we take a lot of copies of these probe molecules, fix them on flat surfaces like this, like a glass surface or like a slide or blotting paper or anything like that. Okay. But we should note that the probe molecules are longer. Right? I'm using shorter sequences for my convenience only. Okay, then we can go to the laboratory and extract the RNA molecules from the tissue of our interest. They will be in solutions. We don't know which mRNA molecule is present in how many copies in each solution, the solution of RNA from normal skin or that from the cancer skin. But I can label them differently. So that coloring helps me to always note which, even if I mix them, I can track which mRNA has come from normal skin and which has come from cancerous skin, right? When we hybridize now, when we mix them and pour on the spot, they will compete. The mRNA molecules from corresponding to that gene of XYZ will compete with this probe. The mRNA molecules will be binding, something else may also bind, but when I wash, they will all be non-specific binding will be removed, only the specific binding will remain. Now I scan and analyze the data. That means I can get the fluorescent intensities at two different wavelengths, one corresponding to the dye which I used for labeling RNA from cancer skin, and the other wavelength for the fluorescence dye that is used for normal skin RNA. And I can determine which one, where the particular XYZ mRNA is expressed more. In this case, it seems to be more in cancer condition. I ask this question for more genes, we will look at that. It is possible, but a quick recap first. We designed the probes, we created the probe spot, we extracted the mRNA, we labeled, we hybridized on the spot and washed it and then scanned. Then we analyzed the data and concluded. I can do the same thing if I can take more number of genes also. Let's say 3 genes are there, ABC, BBC, CCD. I have to repeat probe generation and spot generation just like before, and I can make 3 different spots like this. I go back to the lab, extract the RNA, label them, mix them, hybridize, and scan. So when I scan, the situation in each spot could be something like this. I wouldn't know until I scan. Once I scan, I get these type of images. When I get the fluorescent intensities for 2 different wavelengths, I get the data and I can put them for ABC, it may be something like this, more intensity at wavelength 2, and for BBC and similarly for CCD. So like this I can get the data. I have to interpret this now. How do I do the interpretation? It goes something like this. The expression status of mRNA, I can say the mRNA is present, mRNA for ABC gene is present in both conditions. For BBC, it is present in both conditions. For ABC, CCD, however, it is probably absent. Okay. And at the gene level, I can say the ABC gene is upregulated in the cancer condition. For BBC, it is undifferentiated, and then I can say it is downregulated, CCD is downregulated in the cancer condition. I can now repeat the same experiment for all genes also. I just have to repeat this for 24,000 or whatever number of genes I have in mind and produce an array with lots of probe spots like this. So for example, I could have gene 1 here, and gene 2 could be represented by probe spot 2, etc. So, and each spot would have the probes the way I described it before, and there may be many genes like this. For example, I might have gene number 437 somewhere here, etc. That's it. So if I can have an array of all the genes, I can repeat the experiment and detect the expression level of all the genes in two conditions. Now, it is possible to create microscopic arrays of probe spots. And that's what it is. Microarrays are DNA probe spots at a microscopic scale in an array format. So let's see what happens when we do the experiment. So we have an array now, microarray, and we go to the laboratory, get the RNA, label them differently, mix them, hybridize them, and when we scan, we get these color images. So just like before, maybe for example, this is gene 2 here with more red, the sec1 intensity color. I have more expression in the cancer condition, whereas this gene in A1 is probably expressed in more in the normal condition. So this is downregulated in cancer condition, whereas what is these spots, the genes corresponding to these spots are undifferentiated. What is in gray is not at all expressed in both conditions. It's not expressed in the skin. So like this, I can perform experiments in microarray using 2 dyes, but I can also do it with only 1 dye, but using 2 chips. This is another variant, important variant of microarray experiments. Here we actually use only 1 dye to label the mRNA from both sources. So when we hybridize, mix them, what the results we will get, and we analyze in a different way. The mRNA that has come from 1 chip corresponds to the normal skin. So I compare A1 with A2, which has come from hybridization with a different source. So A1 will be compared with A1 here, A2 will be compared with A2 here, A3, etc. That's it. Everything else is similar to the previous experiments. Now, I will try to make more videos like this. My intention is to bring complex concepts in simple way if possible in the first 2 minutes, and tell some more details later in the videos I make. There are a lot of questions about microarray every beginner should know. One of the most important question is microarray, is it relevant today also when NGS and other technologies are there? So I will make videos about each of these questions hopefully soon. This is my 2-minute tech intro series. I hope you will like all of them, and I hope if you like this video, please click on it and share with others. Thank you very much.